This split-screen chart tracks the footprint of the Mexican axolotl. On the left, biomedical research citations reflect one of the most studied models in modern science. On the right, wild population numbers plummet from 6,000 per square kilometer in 1998 to near zero today. In the laboratory, Ambystoma mexicanum is prized for its capacity to perform true epimorphic regeneration. When an axolotl loses an appendage, it does not form a scar. It perfectly regrows the amputated tissue, bone, and muscle. This capacity extends far beyond limbs. Crush a spinal cord, excise heart tissue, or remove portions of the brain, and the axolotl will restore the missing structures with morphological and functional fidelity. To accomplish this, the organism relies on an extreme genetic architecture. The axolotl genome spans roughly 32 billion base pairs, nearly 10 times the human genome. It also exhibits obligate neoteny. The animal completes its entire life cycle, reaching sexual maturity and reproducing, while physically locked in a larval state. Yet, despite being bred by the millions for research and the pet trade, the species is functionally collapsing in its native habitat. To extract the blueprint for scar-free healing, we must deconstruct its biology across 3 descending scales: its bloated genetics, its arrested hormones, and its precise cellular mechanics. Because of its prohibitive size and complexity, the axolotl genome remained undecoded until 2018, when long-read sequencing technologies finally allowed scientists to assemble the sequence. This timeline represents a standard human genome scale, but the axolotl's 32 gigabase blueprint dwarfs it by a factor of 10. This genetic bloat is driven primarily by long terminal repeats, or LTRs. These repetitive sequences multiply and expand the spaces between critical genes. These repetitive elements account for up to 70% of the entire sequence. They create massive, elongated introns and intergenic spaces throughout the DNA. This architecture is not evolutionary junk. The expanded spacing actively influences how chromosomes fold and conform during tissue repair. By looping into these highly specific 3D structures, the genome enables long-distance regulation of genes, orchestrating the complex mechanics of regeneration. Analysis of this sequence revealed that the axolotl completely lacks PAX3, an essential developmental gene that regulates muscle and neural formation in all other known tetrapods. The axolotl has entirely outsourced these critical developmental functions to a paralog gene named PAX7. The axolotl's enormous, repetitive genome serves as a specialized spatial blueprint. The physical folding of this sequence allows genes like PAX7 to drive prolonged cellular proliferation in the wake of an injury. At the organismal level, the axolotl relies on obligate neoteny, the ability to reach full sexual maturity without ever undergoing a typical amphibian metamorphosis. This diagram illustrates the standard amphibian hypothalamic pituitary thyroid, or HPT, axis. Under normal conditions, signals from the hypothalamic stimulate the pituitary to release TSH, which then drives the thyroid to produce the remodeling hormones T4 and T3. In the axolotl, this developmental pathway is bottlenecked. Genetic variations in thyrotropin-releasing hormone and the deiodinase type 2 enzyme interrupt the cascade. This bottleneck severely reduces pituitary TSH secretion, resulting in exceptionally low circulating levels of T4. It is a misconception that the axolotl is entirely immune to thyroid signals. The organism does possess functional thyroid hormone receptors in its tissues. In vivo, these receptors undergo alternative splicing, generating isoforms that lack parts of the ligand binding domain. This acts as a defensive buffer to protect tissues from sporadic hormone exposure. In a laboratory setting, scientists can intentionally bypass this buffer and force metamorphosis by adding exogenous T4 directly into the axolotl's water. This side-by-side model demonstrates the cost of that terrestrial transformation. The forced adult on the right experiences a 2-fold reduction in its regeneration rate, and amputations result in permanent digit and carpal malformations. Developmental arrest is a strict physiological strategy. The axolotl maintains its neotenic state to preserve the cellular plasticity needed to execute perfect tissue repair. Upon amputation, an axolotl immediately initiates repair. The epidermis rapidly migrates across the fresh injury site, sealing the wound without forming any fibrous scar tissue. This marks the onset of epimorphic regeneration, a process that completely bypasses the fibrotic scarring that blocks limb restoration in humans. The axolotl achieves this by utilizing a unique immune response. It limits neutrophil infiltration and delays extracellular matrix production, maintaining an unscarred, flexible environment. In this cross-section of the stump, you can see the initial wound epithelium thickening and transforming into a highly specialized structure called the apical epithelial cap, or AEC. To sustain regeneration, nerve fibers from the severed stump must grow directly into this new cap. Once established, these nerve endings bridge communication with the epithelium by releasing a neurotrophic signal known as the NAG protein. This interaction initiates a cascade of chemical instructions that targets the underlying connective tissue. The mature, structured cells respond by dissolving their boundaries. They de-differentiate, reverting back into a swarming, highly proliferative mass of primitive progenitor cells. This resulting mass of de-differentiated cells is the blastema. To organize this raw material, the blastema deploys FGF and sonic hedgehog signaling cascades, establishing the anterior-posterior patterning of the limb. Wnt/beta-catenin and BMP pathways are then activated to drive proximal-distal progression, building the limb outward with exact morphological fidelity. Epimorphic regeneration relies on a highly orchestrated reversal of cellular maturity. It is dictated entirely by a flawless, nerve-dependent sequence that humans cannot natively replicate. The axolotl's extreme biology evolved in a highly stable, specific aquatic ecosystem. Its native habitat consists of the high-altitude lakes of the Valley of Mexico, an environment historically shaped by traditional Aztec agriculture. Indigenous populations developed the Chinampas system, building artificial agricultural islands that created a perfect network of shallow, oxygen-rich canals for the axolotl to thrive in. Despite an unmatched biological resilience to physical injury, the species exhibits severe ecological fragility and sensitivity to environmental shifts. Today, aggressive urban expansion in Mexico City is suffocating the wetlands. The traditional chinampas are being abandoned or converted into profitable event venues and soccer fields. This unchecked expansion brings severe wastewater runoff, actively depleting the oxygen in the remaining canals. Invasive carp and tilapia, introduced to the lake system in the 1970s, now dominate the waters and relentlessly consume axolotl eggs. The collapse of the wild population represents a deep cultural tragedy for a species embedded in Mexican heritage and mythology as a symbol of national identity. The axolotl possesses a molecular blueprint that informs new approaches to human wound care and tissue regeneration. However, to truly harness biology's most efficient repair system, we must ensure the survival of the fragile ecosystem that engineered it.